So thank you everyone for being here. Good evening, my name is David Israelowitz, and I'm chair of the J. Robert Oppenheimer Memorial Committee. And on behalf of the committee, I want to welcome Professor Raff, who has excellent technical computer skills, and all of you tonight to our 51st Annual JRO Memorial Lecture. I would like to tell you a little bit of our committee and our efforts. Our committee began in 1971 when John Broglio realized more needed to be done to honor J. Robert Oppenheimer, as we all know, the director of Project Y during the Manhattan Project and the founder of modern Los Alamos. Oppenheimer had been humiliated in the throes of McCarthyism. His security clearance was revoked after a contrived and unfair set of hearings where he was forced to defend himself and his reputation after leading one of the great scientific and national security achievements of our time, one of great geopolitical and moral complexity, which he never shied from wrestling with the rest of his life. Eventually, the committee determined that one way to honor and preserve Oppenheimer's memory and his many contributions to science and national security was to organize an annual lecture by an eminent scholar, the latest of which we will enjoy tonight. In addition to these lectures, our committee provides scholarships to promising students from local high schools, thanks to the generosity of local organizations, from bequests, from small and large donations from our donors. This year, I'm very proud to say that we awarded a record 17 scholarships to Los Alamos and regional high schools, which now comes to a total of 308 scholarships since our program began in 1984, bringing our grand total to just over $806,000. More recently, we have begun to encourage our local talented youth at a younger age, giving awards and recognition at science and history fairs and other school-based programs. So it is fitting that we recognize this year's scholarship winners. And we have some of them in our audience. If you are one of our scholarship winners, please stand where your name is called, and please remain standing. And the audience, please hold your applause until we have read all the scholarship winners' names. So the J. Robert Oppenheimer Memorial Scholarship goes to Rebecca Lee of Los Alamos High School. The scholarships in memory of Nicholas Metropolis. Could we raise the lights for just a minute? Thank you. There's Rebecca, just so you know, for future reference. Scholarships in memory of Nicholas Metropolis go to John Cockwell of Los Alamos High School and Joaquin Carabello of Coahque Valley High School. In memory of Nancy Lobach and Art Fried, a scholarship goes to Shiv Antar Khalsa of Los Alamos High School. In memory of Rosalie and Liam Heller, to Daniel Kim of Los Alamos High School. Scholarship funded by the Dell Foundation in memory of Saj Cowan, go to Araya Vigil of Coahque Valley High School. In memory of Terry Hawkins, go to Aliana Cornflower Ortiz of Santa Fe Indian School. In honor of Mary Lou and Michael William, go to Anelia Alterman of Española High School. Funded by the New Mexico Consortium to Chloe Lee of Los Alamos High School and Natalia Marquez of Santa Fe High School. In memory of Richard Swenson to Deanna Foster of Los Alamos High School. By the Los Alamos High School class of 1970 to Emma Frost of Los Alamos High School. And then we're fortunate to provide additional scholarships funded by our committee through our donors that were awarded to Maximus Posada of Los Alamos High School, Añana Martinez of Capitol High School, Merrick Ward Brown of Santa Fe High School, and Dalian Lopen and Mateo Nagel, both of Poaki Valley High School. So congratulations and the best of luck. So why are we all celebrating Avi's legacy here in a small northern New Mexico town? Why is Los Alamos here anyway? Let's look back to 1922, when 18-year-old Robert Oppenheimer was in frail health in New York City. His parents thought sometime in the Southwest would restore him, and by a simple twist of fate, Oppenheimer's rehabilitation began in the shadow of the beautiful Sangre de Cristo Mountains, and he fell in love with northern New Mexico. He always said that his two great loves were physics and New Mexico. When it came time to launch the Manhattan Project, the government looked to the Southwest for a remote site. After rejecting Hemet Springs, Oppenheimer suggested the site of the Los Alamos Range School. General Wesley Groves immediately approved, and the rest, as they say, is history. What was originally envisioned to be a few hundred scientists, engineers, and craftspeople working in isolation has now become the premier national laboratory in the Department of Energy Complex, employing over 16,000 people. And we are delighted that Laboratory Director Tom Mason is here again, available to introduce tonight's speaker. So I invite Dr. Mason to come up. Thank you.
Welcome, and thank you all for being here for this 51st Annual Oppenheimer Memorial Lecture. It's my pleasure to introduce our distinguished speaker, Professor Jennifer Raff, who will be presenting to us this evening on the subject of ancient DNA and the peopling of the Americas. Jennifer is engaged in research at the intersection of anthropology and genetics. Her research interests range from ancient DNA to contemporary genetic diversity. She's also a gifted communicator who's passionate about science literacy, and she conducts her work in anthropology and genetics with a notable sensitivity to the cultural context in which that work needs to be interpreted. At Indiana University, Jennifer studied molecular, cellular, and developmental biology and biological anthropology, earning a doctorate in anthropology with a focus on genetics. She's currently an associate professor of anthropology and an affiliate faculty member with the Indigenous Studies program at the University of Kansas. Working with tribes and communities across North America, Jennifer uses genomic analysis as a tool for investigating historical questions. Her research in this area focuses on the initial migrations of people into North and South America, as well as more recent histories of the North American, mid-continent, and Arctic. In addition to presenting papers at professional conferences, Jennifer has written articles for the public in The Guardian, New York Times, Scientific American, Forbes, and her blog, Violent Metaphors, Thoughts from the Intersection of Science, Pseudoscience, and Conflict. She's the author of an award-winning and best-selling book called Origin, A Genetic History of the Americas. She recently received a prestigious Guggenheim Fellowship to support work on her next book, The Ancients, The Untold Story of How We Became Human. Please join me in a warm round of applause for Jennifer Raff.
Thank you. Thank you so much for that incredible introduction and for having me out here. I've had amazing visits so far. I wish we could stay longer. But I've really enjoyed meeting so many people here, especially the students who I got to have dinner with last night. I want to add my congratulations again. You guys are incredible. They are so smart, so talented. Yes, so I'm really delighted to be here, and I wanted to ask that as we gather today to talk about history, I've lost my cursor, hang on one second. I'm not so good at tech, actually, it turns out. I would like to acknowledge and thank the indigenous peoples whose lands we are currently on, particularly the Pueblo peoples in this region and across New Mexico, or what we call today New Mexico, and their ancestors, and recognize their ongoing relationship with the land. My book was written at the University of Kansas primarily, which is on the traditional lands of the Kaw, also known as the Kanza, and the Osage Nations. And I think it's important that we can understand and we can acknowledge the histories of places throughout the Americas as a step towards equity and justice for past harms, but it's only the first step. And I think that land acknowledgements can actually become kind of performative, right, if they're not backed with more. And members of our research group strive to actually return tangible benefits to the indigenous communities with whom we share space and collaborate, and on whose lands we work and visit. And if you're interested in how we do that, I'm happy to share specifics after the talk, and I'll give you another example at the very end of this talk. I also want, before I begin, I like to do my acknowledgements at the beginning. I want to acknowledge and thank the Indigenous and non-Indigenous scholars whose work has informed my thinking on these issues. And I want to stress that much of what I'm going to be talking about today is not necessarily my own research, but it's actually the scholarship and the academic and the public scholarship by many of these scholars. They're Indigenous scholars and non-Indigenous scholars. And these are some, but by no means all, of the people who have influenced my approach to research. Okay, that is a very big picture. It didn't look this big on my screen, sorry. So when I was first invited to give this lecture, I was really honored and really excited, but I was also a little bit puzzled because I didn't really know what the relevance of my research and writing, which is primarily concerned with archaeology and genetics, what that is, how that relates to the work of Robert Oppenheimer. And I didn't know much about him, beyond a little bit, so I've spent the last few months learning about him. And I have by no means an Oppenheimer scholar at this point, and in this audience, I know there are some, so you can please tell me later what I get wrong. But I have come to realize through my writing and my reading that there's actually more connections between my work and his work than I would have guessed. This is not a very unnatural pairing. So one of Oppenheimer's legacies was his support of scholarship outside the realm of physics. He supported the arts, the humanities, the social sciences, and even the mystical. These all commanded his deep respect and his fascination. He spanned multiple disciplines and encouraged out-of-the-box thinking. And I think that's another major point of connection between the study of the origins of humans on all continents and Oppenheimer's legacy. And as you will see, the story that I will tell you today, this is one small piece of human history, but the quest for understanding human history makes use of multiple lines of evidence, and it really cannot be fit into any neat disciplinary boundaries. I think, as you're also going to see, at least as I was working on this talk, one of the through lines that I discovered linking the deep past, the early history of archaeology, and the more recent history of Oppenheimer's work, and our presence here today, right now, is the land, which is known today as New Mexico. And you'll see, as I go through this talk, how often New Mexico comes up. So I'm going to be talking today about the very distant past. These are events that occurred over 15,000 years ago. And it's worth taking a moment to think about how we study the past. There are many approaches to this, and none of these approaches gives us a complete answer by itself. So many Native American societies have histories of their own origins, which have been transmitted, in most cases orally, for many generations. Some of these traditions and some of these histories align with scientific ways of studying the past, and some do not. My personal focus, my research is on other ways of studying the past, particularly archaeology and genetics. But I want to take a moment to respectfully acknowledge the many ancient perspectives on histories, some of which are understood to be metaphorical, and some of which are taken as literal explanations of origins. I am not telling those stories because they're not mine to tell, but I strongly believe that they are important. Events in the past unfolded in particular ways, of course, but our ability to understand what has happened is limited by the tools we use and the data that are available. And it is worth noting that far from being perfectly concordant, the stories that archaeology and genetics tell, which I'm going to be speaking on primarily today, they often seem to be different. Archaeology seems to tell us something different than genetics, at least in this present moment. So this is all to say that while I prioritize genetics as a tool for understanding the past in my own research, the stories that DNA tell are not complete, nor are they the only way to understand how the past evolved. And the same is true with all of these approaches. So today I'm going to be talking to you about the latest scientific understandings for how the first peoples came to the Americas. These were the last continents to be explored by modern humans. These were the last step on our global journey. And there are so many questions about this process. Who were they? How did they survive the harsh conditions of the last ice age? Why did they travel? How did they adapt to the new lands and the new environments that they encountered? We once thought, we being anthropologists, that we had a pretty good idea of how this process unfolded. But we've had to revise our models many, many times in the last few decades, and we're going to go through that in this talk. Thanks to the introduction of a new kind of data, complete genomes from people, we have learned in recent years that this story is far, far more complicated than we ever envisioned. We are learning so many new details each year that it is impossible. It's really hard even for me to keep up with this, and that's my job. It's very hard for people who are not steeped in the literature every day to keep up. So although the story that I'm going to be telling you today is going to be changed in a year, 5 years, 10 years, what I'm hoping to do today is to give you an accessible summary of the state of the science so that when you hear about these new results that will be coming out in the future, you'll be able to contextualize them. But as we go through this talk, there's going to be another theme, which is the way that we have collected our data, we scientists. And it is very, very important that this is woven throughout this story. There have been many nefarious attempts to use stories about Native American origins as tools for disenfranchisement, for land theft, for genocide. We have to be mindful of this history, although it's not something I can do justice to in this short talk. It's a whole different talk that I could give, but I'm not doing that tonight. So let's keep that in mind as we go through this. So I want to start our exploration with a bit of archaeological history by taking you back in time to the year 1908, to the Crowfoot Ranch near Folsom, New Mexico. I'm sure some of you guys are familiar with this. It's September, and the air is hot, and the cowboys riding through the Wild Horse Arroyo are looking for stray cattle after a major flood nearly wiped out Folsom. This man, George McJunkin, his attention was caught by a pile of bones at the base of the arroyo. He recognized at once that they could not have belonged to one of his stray cows. These were very old bones, not freshly killed. And he also recognized that they were not the bones of cow. They were actually the bones of a bison, and they were not the bones of any bison he had ever hunted before. They were something new that he hadn't seen before. After closer inspection, he removed some of the bones from the site and began to try to rouse interest from other people in order to investigate this find. And McJunkin's recognition of the importance of these remains was a reflection of his past, his experience as a cowboy, as a bison hunter, and as a self-taught, well-educated naturalist. McJunkin had been born into slavery in Texas, and he'd been living as a free man since the end of the Civil War. He left home to take up the life of a cowboy, and he went on cattle drives, and he traded his expertise in horsebreaking for reading lessons. His knowledge and his experience and his fascination with science and the natural world meant he was the right person in the right place at the right time to make one of the most pivotal discoveries in all of archaeological history. So the Folsom site, as it eventually came to be known, would reveal to archaeologists the remains of dozens of extinct bison, and many of them were associated with spear points like the one seen in this photo. This was a paradigm-shifting discovery, which established that humans had been present in the Americas during the Pleistocene, rather than being latecomers within the last 5,000 years, which is something many scholars believed at the beginning of the 20th century. This is just one of a number of paradigm shifts in scientific understandings of the origins of the first people that we're going to discuss in this talk. The Folsom site not only presented archaeologists in the early budding field of archaeology with this evidence and this answer to this debated question, but it also taught them how to locate early sites in the Americas based on their appearance. And so by searching for the remains of extinct animals, archaeologists began finding more and more evidence of the humans that had hunted them. Following the discovery of Pleistocene age artifacts at sites like Folsom, they began to find more and more of these early sites. And as radiocarbon dating made it possible to establish absolute chronologies beginning in the 1950s, a new model to explain the peopling of the Americas began to take shape. And this has come to be known as the Clovis first model. If you're my age or older, you may have learned this in school. So this model took into account the apparently sudden and widespread appearance of people who made the Clovis technologies, which are characterized by this bifacially flaked fluted stone point that you can see right here. And it was found first at the Clovis site, again, a really nice connection. And these appeared very abruptly, seemingly, in the archaeological record across North America around 13,000 years ago. This is a map of the distribution of fluted points in North America. It's incomplete, but you can get an idea of how widely distributed these Clovis points were. These sites were taken as evidence of the first peoples of the Americas, but how did they get here? So the environment of the late Pleistocene can provide us with some important context. This is a GIF. It should be running, I hope. Yes, okay, good. So the region that we're going to be focusing on is called Beringia, which constitutes the lowland regions between Siberia and the Mackenzie River in Canada. And this region remained ice-free during the last glaciation. So the environmental conditions during the last ice age, or as we call it professionally, the last glacial maximum, it was a period of global cooling and arid conditions that reached its peak between about 26,000 and 19,000 years ago. And these environmental conditions play a key role in understanding the peopling of the Americas. So during the LGM, I'm going to abbreviate last glacial maximum as LGM, think ice age when I say it. During the LGM, enormous quantities of water were bound up in glacial ice that covered enormous stretches of the continents, including basically most of North America, not Alaska, but most of North America. This meant that the coastlines were very different than they are today, and ocean levels were approximately 100 meters on average lower than they are today. So this meant that an enormous land connection between Asia and North America existed, and this is known as Beringia. You can see a GIF of the coastline changes here in the timeline beginning about 21,000 years ago. So the LGM was a very harsh time in which to live. So besides being bitterly cold, it was so dry that large regions of the northern hemisphere were actually desert. This affected the distribution of plants, which affected the distribution of animals, which in turn affected the distribution of humans. And around the world, the archaeological record shows us that at this time, people retreated into climate refugia. The conditions of the LGM also prevented people from moving very far into the Americas. So they could cross Beringia through this land connection, the Bering Land Bridge, between 50,000 and 11,000 years ago. But they could not move out of present-day Alaska because the way was blocked by a massive ice sheet that was formed by the joining of the Laurentide and Cordilleran glaciers. Potential routes of entry began to open up slowly as the Earth warmed after the LGM. And the first was along the Alaskan coast, by about 16 to 17,000 years ago. And the second was in the interior of North America. And the estimates for when this interior route first became accessible are a little bit controversial. We're not really sure about that. But we know that it was definitely open and there were animals traveling through this land connection, or through this pathway that was opened up between the ice glaciers by about 13,000 years ago. So the Clovis first model suggested that the ancestors of people who made Clovis sites came from Siberia, where they led a rugged, manly, it was very gendered back then, manly existence, hunting these ice age beasts like the mammoths. And they lived in small extended families and bands that ranged widely around the continents. And these ancient Siberians crossed the Bering Land Bridge following the mastodon herds, and they moved swiftly down this ice-free corridor, peopled North America very quickly 13,000 years ago. So this was an elegant model. Like I said, you probably learned this in school if you're my age or older. Most archaeologists agreed it accounted for nearly all of the archaeological evidence. And it persisted for about 50 years. It was so entrenched in the archaeological community that it really dominated the field for decades. But evidence kept appearing that didn't quite fit the model. So even as better dating methods pinpointed the earliest appearance of Clovis to about 13,200 years ago, the Clovis first model still couldn't satisfy everyone. From time to time, a maverick archaeologist would propose a site that didn't fit the model and was older than Clovis. And they were pretty roundly criticized, that this was a really entrenched theory. I also want to take a moment to point out that the indigenous peoples across the Americas themselves have steadfastly maintained that they have been in their lands a lot longer than 13,000 years, so we should note that. However, at one point, some archaeologists came together and agreed that, let's see if I can point this here. Wait, I've got a laser pointer. This site, right here, down at the bottom, Monteverde in Chile, actually was legitimately pre-Clovis. It dated to 13,400 years ago, I think, and I forgot. Anyway, it's quite old, and it was pre-Clovis, and what's more, it was in South America, so this could not be explained by the current models, right, of peopling the Americas through this corridor. This is kind of a list of some selected, oh, I'm sorry, Monteverde is 14,200 years ago. Oh, don't tell Tom Delahaye, I'll get in trouble. Actually, he thinks it's older than that, but we won't get into that. These are a number of the major pre-Clovis sites that have been accepted by many archaeologists, not all. There are a lot that are missing from this map, I'll just say. But in any case, this represented another paradigm shift in archaeology. Archaeologists began looking at these pre-Clovis sites and recognizing more and more of them elsewhere. Okay, so I want to turn now to genetics, and I call myself an anthropological geneticist, which is not really a household term, so I can define it really quick for you. Anthropological genetics is a synthetic field which applies the method and the theory of population genetics to evolutionary and cultural questions of anthropology. And in our field, it's been very exciting because over the last decade or two, genomic approaches have been increasingly common in investigating human population origins and population histories and biological relationships among diverse human groups. And we are working in that area. Anthropological geneticists work on all aspects of human and sometimes non-human genetics, but in general, the field addresses two main questions. How do humans differ biologically, and how have aspects of human histories shaped these biological differences? But it's important to be very clear about what DNA can and cannot tell us. So DNA can give us estimates of past population sizes with some caveats. It can give us information about sources of ancestry. It can give us information about demographic events, so migrations, gene flow between populations. Those leave very discernible signals in genomes. It can also give us approximate dates of these events, again, with caveats. What it cannot tell us, though, aren't necessarily are the location of events, unless it's tied to archaeological evidence. It cannot tell us the difference between two populations if they are or were very genetically similar. It cannot tell us about identity or behavior. And it does not tell us anything about tribal affiliation. That is a totally different question. That's a totally separate set of issues, not steeped in DNA. So some of the people in this audience have backgrounds in genetics and some do not. So I want to really, really quickly go through a little bit about DNA and just to get us all on the same page. And I want to ask your pardon in advance for my really terrible cartoons. I am not a graphic designer. Yes, this is supposed to be a cell, and that circle in the middle and with those X's, that's supposed to be a nucleus. Those of you who are geneticists will recognize that these are chromosomes in replicated states, and there's not enough of them, but we're not going to worry about that right now. And these are supposed to be mitochondria. Okay, so when reconstructing population histories, geneticists can rely on different aspects of our genomes. So mitochondrial DNA, abbreviated as mtDNA, is the DNA present in the mitochondria of your cells. Those are the energy-producing organelles that are within your cells. This is a separate genome from that found in your chromosomes in the nucleus. Mitochondrial DNA is maternally inherited, and it doesn't swap genes in and out like your chromosomes do, or it's called non-recombining. And it's present in hundreds to thousands of copies per cell. These factors make it really great target for ancient DNA research. And we're going to talk about this shortly, but ancient DNA is fragmented and scarce. And so it's really nice to have a source of DNA that is abundant, relatively. But so many, many ancient and modern DNA studies are based on mitochondrial DNA, and it does give you a great idea of population history, but it only gives information about a tiny subset of ancestors, your mother, her mother, her mother, and so on. So it gives you an accurate but incomplete and low-resolution glimpse of history. So I liken this to the edge pieces of a puzzle. You can maybe figure out what the picture is, but you're missing a lot of details. To get a more detailed picture, one needs to examine nuclear DNA, which is the genome present in your chromosomes. So in contrast to the 16,500 A's, G's, T's, and C's that are present in your mitochondrial genome, the nuclear genome contains about 3.1 billion bases, and that's the haploid, the half of the nuclear genome, like one set of chromosomes. So that's about 30,000 genes. Unlike mitochondrial DNA, nuclear DNA is present in only 2 copies per cell, so one from mom, one from dad. This means it's much less abundant in ancient human remains, and it's much more difficult to retrieve. But when you can get it, it is a vastly more powerful tool for population genetics because it is inherited from both parents, and it reflects the history of all your ancestors, not just the ones on your maternal lineage. So it provides you with millions and millions of pieces, puzzle pieces, including the middle pieces, that allows you to reconstruct a highly detailed picture of human history. And what's really exciting about the age we live in now is that we can get these puzzle pieces from ancestors as well as living peoples today. And this field is called paleogenomics. So I've mentioned ancient DNA a few times now. I want to note that, and I've noted that the study of ancient DNA has been crucial in discovering our understanding of the earliest peoples of the Americas. It is an extremely valuable tool because it gives us a direct window into past populations. Using ancient DNA, we can say, at this time, dated using radiocarbon methods, we know that these genetic variants were present in this person at this geographic location. But it is very, very difficult to recover. It is degraded, it is scarce. The mechanisms for repairing your DNA that are going on right now as you're sitting here, you're generating mutations and your body is repairing them, we hope. Those repair mechanisms stop once you die, and your DNA begins to degrade. And the rate of degradation depends on the environmental conditions that you are in. So we can recover DNA much more easily from cold and dry environments than we can from hot and wet environments. And so it is highly, highly variable. It's also extremely sensitive to modern contamination. And that means there are very many fewer genomes that are recoverable to work with. So aside from these exceptional cases that you read about in Nature and Science, the majority of ancient genomes are very poorly preserved, if they're preserved at all. Most of them are not. And it is very technically challenging to take the precautions necessary to ensure that DNA from an ancient sample of an individual is uncontaminated. This is our lab. This is one of the grad students, I don't know who that is actually, trying to get DNA from a soil sample. So paleogenomics has been gaining an incredible amount of attention in the media and in scientific journals in recent years. The Nobel Prize was awarded to Svante Pääbo, who actually was one of the founders of our field and sequenced the Neanderthal genome. I get to meet him in September. I'm really excited about that. In the Americas, paleogenomics researchers have addressed different aspects of population histories. Some of the most visible papers in recent years have focused on the initial peopling of the Americas. But there are other researchers, including me, who are looking at more recent aspects of population history. As of 2021, this figure is already out of date, these are the number of individuals from the Americas whose genomes have been sequenced. And these are, I believe, these are all ancient individuals. So here, these are the number of individuals, and the circles are proportional to the number of individuals from that region. These are where we have genomes from between 12,800 and 8,000 years before present. Not very many. Here in the middle, we have between 8,000 and 4,000 years before present, and then less than 4,000 years before present. That's what we've got. Again, it's a little out of date, but not super out of date. I want you to notice the gaps. It's very important, okay. But before we get to the gaps, I want to talk about what do these genomes actually tell us about biological history. And again, caveating this is not telling us about cultural history. This is not telling us about indigenous people's own histories that they have maintained for millennia. This is telling us about biological histories only. They're a complicated story. So all of the complete genomes, ancient genomes recovered from the Americas, show us that they have two major ancestry components. The majority of their ancestry comes from the ancient peoples of East Asia. We know that from, well, a number of genomes from this region. The other major ancestral component was discovered through the sequencing of the remains of a little toddler who lived 24,000 years ago at the Upper Paleolithic site in Siberia called Malta, right here. Archaeologist Kelly Graff, who is one of my colleagues at KU, University of Kansas, was instrumental in arranging for the genomic study of this child. And his genome shows us that his population intermarried with a group of ancient East Asians sometime between 25 and 20,000 years ago. There's a lot of fuzziness associated with these dates, so they may change. But out of this meeting and intermarriage emerged the ancestral group that gave rise to multiple populations, but including Native Americans. And I've marked that, let's see, right here, Beringian ancestors. They also gave rise to Paleo-Siberians. I'm not going to talk about them today, but we're talking about the Beringian ancestors here. And so this is a tree kind of showing, this is meant to depict gene flow, producing present-day East Eurasians and the ancestors of Beringian populations about 22,000 years ago, give or take. We also see evidence for an ancestry component in some Amazonian groups that is related to present-day Australasians. This is known as population Y, and this is a really fascinating puzzle to geneticists. So we can rule out, for now anyway, the most obvious explanation that probably jumps to your head right away, which is a trans-Pacific migration from Australia to South America. For technical reasons, which I am not going to get into right now, but I'm happy to talk about later after the talk if you would like, that does not fit the model that we would expect. The patterns of that, the model that we would expect to see for that to be the case does not yield the genetic variation that we see in their genomes. This is a much older signal. It's much more ancient. And in addition, this ancestry has also been found in this individual here, Qianwan man from 40,000 years ago from East Asia. So there are two kind of models that might explain this, that fit the data. One of them is that both population Y and contemporary Australasians population-wide being that name that we give that ancestry signal that we see in South Americans. I can't remember if I told you that already. This idea is that both of these populations descended from an ancient group widespread through Asia and mainland Asia, and that the Tianwan individual was closely related to that group, and that ancestry just kind of moved in different directions as people move around. That's quite plausible. Another possible model, which we cannot discount yet with current evidence, is that there was a very early migration of people bearing that ancestry signal early, before the last glacial maximum. And I'll return to that question in a minute. So the ancestors of the first peoples, following the mixture of the ancient North Eurasians and the Eastern Eurasians, they mixed together. And then there's this population that I'm designating Beringian ancestors. This group of this population remains isolated at the peak of the last glacial maximum for several thousand years ago. And during this period of isolation, they evolved genetic variants that we see only in present-day Native Americans. So I want to take a second to talk about this isolation because it's really interesting. This is sometimes referred to as the Beringian pause or the Beringian standstill. I don't know that either of those terms are particularly useful, but many of us think that this, I told you earlier we can't tell geography from genetics, and that remains true. But many of us think that this period of isolation took place in Beringia itself, perhaps along this southern coast, because paleo-environmental reconstructions have shown us that this was actually a relatively decent place to live compared to everywhere else in the northern hemisphere during the peak of the LGM. The temperatures were warmer, there was more water, there were more things to eat. But we don't have any actual physical evidence of people being there. There have been a couple of sites proposed down here that maybe date to that time period, but they're very controversial. It's not clear that they're actually archaeological sites. And we know that people were up in Beringia as early as almost 32,000 years ago from this site, the Yana rhinoceros horn site. There's very clear evidence of humans living there. So we know humans were above the Arctic Circle by 32,000 years ago, give or take. We just don't know if they were right here because this area is now underwater and we cannot access it to explore it archaeologically. Okay, so as this population that was ancestral to Native Americans was isolated, our best genetic models show us that it was splitting into several subgroups. And we know this from the genome recovered from two individuals, one here at the Upward Sun River and one here at the Trail Creek Cave site, that show us back into history. So these are kind of recent ones, 9,000 years, One is 11,500 years ago, but they tell us about their ancestors back in the past. And it shows us that, I'm gonna go back one more. It shows us that one branch has been referred to as ancient Beringians. I don't like that term because it gets very confusing with these labels, but this population was not ancestral to other Native Americans. It remains isolated in Alaska, and that's where these two individuals belong to this population. Another population split off about this time, and it's kind of referred to, not a great term, as unsampled population A, because we don't have any genomes. We have not sampled any genomes directly from this population. It has been inferred by its contribution to the DNA of present-day Mixe peoples of Central America. We don't really know a whole lot about that. And then the last branch moves south of the ice sheets and becomes ancestral to all other Native Americans. So how does that happen? And this is kind of the last phase of the peopling of the Americas, right? How do people move south of the ice sheets? Well, genetically, we have some clues as to, remember I told you about two different routes, right? One was possibly this coastal route that people might have taken. Another was this interior route. Genetics show us that it was more likely that people took the coastal route than the interior route. We see the subdivision of all Native American genomes into two branches. One has been referred to, again, these names are not great, as Northern Native Americans, and that refers, people descended from the Northern Native Americans, or NNA branch, include Algonquians, Na-Dene, Salishan, and Tsimshian speakers in Canada, and everybody else, including North Americans, at least as far north as Montana, everybody else belongs to this branch called Southern Native Americans, or SNA. Okay. And these two branches give us a hint that people were south of the ice sheets pretty early. These two groups separated from each other between about 17,500 years ago and 14,600 years ago. That separation, usually, those kinds of separations usually occur as people are moving, right? And populations are splitting from each other. And it is thought that that separation happened south of the ice sheets, and it really fits more with a model of an early migration down the coast because the the interior route was not open in time for that migration, if that makes sense. So this is a model kind of showing these two possible migrations. Here's the ice-free corridor, right? And in addition to not being fully open in time to produce these other archaeological sites that predate 13,000 years ago, it doesn't fit with that split of these two major genetic groups. Instead, probably what happened, at least the best models that we have right now, suggest that people moved rapidly by boat along the coast and moved very, very quickly down the coast and peopled the Americas that way. We don't have any direct evidence of this. We don't have any evidence of boats, right? We have nothing like that directly, but the genetics suggests that that's what happened. So it needs further testing with additional data, as do all things in archaeology. In recent years, and yes, this is just an excuse to put up puppy pictures. I will admit this, I'm sorry. I'm not sorry, I do it. In recent years, we've been able to get insights into the human history from unexpected sources, and that includes the genomes of dogs. So humans and dogs have a very ancient relationship. They were the first animals ever domesticated. They were actually domesticated up in the Upper Paleolithic. We have co-evolved as domestic partners for thousands of years. And genetic evidence shows us that American dogs did not evolve independently from American wolves. Instead, they show a clear signal of descent from Siberian wolves, suggesting that they were brought to the Americas with the first peoples. And the remains of dogs can be seen in archaeological contexts, at least by 10,000 years ago, if not sooner, or if not earlier. So an understanding of the dynamics of dog population history can give us insights into how people moved as well. And there's a recent study by Angela Perry and her colleagues which looked at mitochondrial DNA, right? We talked about that at the beginning. They looked at mitochondrial DNA of dogs and looked at their branching patterns. And what they show us is there are some clades that remain in Northeast Asia, but there are some clades that move into North America and begin to branch out. These branches begin diversifying sometime after 15,000 years ago. If you compare that to the genetic evidence of human populations, you see that these match human population splits. I mean, you may not be able to see the details, but I think you can get the idea of the branching patterns here. And these nodes show us the approximate dates of these branches. And you see they line up really well with dogs. So this is really interesting. We would really like to see more data from dogs. Complete nuclear genomes would be great. We're waiting for that. I hope that will come soon. But for right now, the dog migration patterns seem to mirror genetically human migration patterns, and that is very cool. Okay, so I'm going to try to sum it up as far as dates go, because I just threw a whole lot at you at once. So when we're talking about when did the peopling occur, I'm going to give you what the genetics show us right now. So first, the first event is ancestral Native Americans diverging from their Siberian and East Asian populations about 25,000 years ago, give or take. These Native American lineages during this period of isolation begin to split between about 22 and 18,000 years ago, and the Native American lineages diverge into two branches, presumably south of the ice sheets, northern Native Americans and southern Native Americans, between about 17,500 and 14,600 years ago. So these are the dates estimated from DNA. They rely on estimates of mutation rates and a whole bunch of other technical stuff I won't get into. These are highly dependent on some assumptions, but they're pretty consistent with each other. Okay, so this was a neat story, but it's really complicated by some archaeological evidence that has occurred recently. So just 2 years ago, a site was published from New Mexico that was evidence of human footprints. And these human footprints span over 2,000 years. So people were at this site laying down trackways for 2,000 years. These footprints have been dated to between 23 and 25,000 years ago, which is the height of the last glacial maximum when, according to all other archaeological evidence and genetic evidence, humans should not have been in the Americas. So this is the White Sands site, specifically White Sands Locality 2, and the exact location of this within the park is secret, but it is still being excavated today, so that is very, very exciting. We're going to learn a lot more. We know that these footprints were made mostly by young people, and I would love to tell you more stories about them because they're fascinating, but I have to keep this talk moving or you're never going to get out of here. But they tell some really amazing stories about the lives of people back that long ago. There was a little bit of, actually there was a lot of controversy when the footprints were first dated. They have since been redated with independent methods. It looks like it's holding up. I don't believe all archaeologists have accepted this because it's a pretty radical paradigm shift to imagine people being in the Americas at a time when those ice sheets are blocking the entrance into Americas. What that means is people would have had to have been in the Americas, they would have had to come into the Americas no later than 25,000 years ago because that's when those ice sheets fused. So people came in very early if the White Sands site is true, and I think it is. I mean, I'll go on the record as saying I really think this is incredible. So we now have a discrepancy between the archaeology and the genetic data. How do we resolve it? I don't know. We will hopefully be able to come up with more models. We'll be able to come up with more evidence, but right now, this is the state of the field as it is today, and it's very exciting to be working here. So, broadly speaking, there are currently several perspectives among archaeologists on the peopling of the Americas. Which one you favor depends on what kind of evidence that you prioritize. So one group of archaeologists still maintains that people arrived in the Americas rather late, maybe just about 14,000 years ago. Another group of archaeologists on the complete opposite end of the spectrum interprets the evidence as showing people were present in the Americas very early, and some even say as early as 130,000 years ago. I'm not really on that board with that. I don't think the evidence is there. That's a very small group of archaeologists, but I'm putting it out there because they're out there. By far, the majority of archaeologists and geneticists interpret the evidence as showing that people were present in the Americas by 16 to 17,000 years ago, or perhaps by 25,000 years ago. And of course, tribal oral histories give many, many answers to questions of their origins. Some of them align with some of these models, and many do not. So there are a lot of perspectives on this. One factor which is complicating the genetic models a lot is that we don't have that many genomes from ancient peoples in North America, especially in the present-day United States. So again, I asked you to look for the gaps. They're there. And this is for a very good reason. So the history of anthropology, of archaeology, of genetics, and scientific research in general, to be honest, is one that is unfortunately a history of abusive research practices towards indigenous peoples. And there has been a perspective among some non-native researchers that advancing scientific knowledge is more important than anything. And that the ends justify the means. And this, along with a lack of cultural understanding of and respect for sovereignty, particularly in genetics, has caused many incidents of harm. And so, as a result, broadly speaking, there's a very understandable lack of trust in scientists, and that scientists will do research in ways that are respectable. I want to stress, though, that Native peoples are not anti-science at all, but there is this lack of trust here. There are also a lot of other issues, particularly in the realm of paleogenomics. So many indigenous peoples hold very strong beliefs that their ancestors' bodies are sacred, that their bodies are sacred. DNA retrieval is not an option, and that there may be tensions and harms that may result when genetic studies and traditional knowledge systems conflict. And there's a bunch of others that I'm not going to have time to get into. Again, this is a whole other talk that I could be giving. It's a very complex topic, but I want to stress that every tribe and nation and community has their own perspective on this. Some, the ones I work with, are okay with doing research, paleogenomics research. Some have restrictions on it, and so it's very, very diverse, and some will just not do any. Many indigenous geneticists and non-indigenous allies have been working together for many years, in recent years, to provide guidance for researchers that acknowledge the harms that have been done, that stress how there is no single one-size-fits-all approach to doing genetics research. But we see cases of successful research, and I try to highlight them in my book, when scientists respect tribal sovereignty, when they listen when tribes say no to research, they respect that. When they work in partnership with tribes and communities who do wish to use DNA for exploring their histories, and when they do so with care and respect for their priorities and concerns. This is a very, very important discussion that is going on in my field right now. Okay, so I want another giant face. I want to return to Oppenheimer here at the end to acknowledge another connection between this field and him. So Oppenheimer, as you know probably better than I do, was a complicated man, and his life embodied many contradictions. But I think many of you as well, since we're here today, admire his legacy as a public scholar and his concern with the consequences and the potential harms of scientific research. I am not drawing necessarily a parallel between the harms caused by the harnessing of nuclear energy and genetics. That's different scales, right? But we must definitely consider the ethical aspects of all scientific endeavors, and this is very true in my field. So the story that I've told you today is not one of the far distant past, it's also the story of the present and more recent past. So how we have come to our scientific understandings of the origins of indigenous peoples is just as important as what we think we know. And this history is, and we have to acknowledge this, deeply rooted in colonialism and centuries of exploitation of indigenous peoples. The results from our research today have the potential to deeply impact Native communities. And we can choose to do this research in a positive way, but only if we have an understanding of the ways in which previous work has harmed Native Americans and ways in which good work has been done too. I don't want to be completely negative here. There are some really great examples of partnerships with tribes that have yielded amazing research. I wanted to write a book that helped people understand just how complex and relevant this fascinating story is and why it matters. That's my book. I have to put it out there so you can see. We've barely scratched the surface of the many different topics in it, but if you're interested in delving deeper, you can read it. And these are some ways to follow me in my work. And this is where I return to the ways in which my research team and I try to go beyond land acknowledgements. We are currently supporting the Mescalero Apache Tribe's Fire Relief Fund to help evacuate and support thousands of people who are being affected by the South Fork Fire and the Salt Fire. And if you are so inclined, I would love to invite you to also support this. You can just scan it with the QR code right there with your phone. And that is my Twitter if you want to follow, although I'm not tweeting as much anymore. Twitter's a mess. That's my website there. Okay, and I'm going to end with this quote from my colleague, geneticist Dr. Justin Lund. He gave this talk for the American Association of Biological Anthropologists in 2021, and he likened the disciplines of genetics and archaeology to houses with crumbling foundations. And he advocated for foundational repair by mending relationships with indigenous groups. And he said, creating bad relationships took generations, and mending those relationships will also take generations, so plan accordingly. If your work starts and stops in the lab, you're doing it wrong. And I think this is a sentiment that I believe Robert Oppenheimer would have agreed with. Thank you.
We have a little surprise for you.
There's more.
The first surprise is, and I apologize to the audience,
We're not going to be taking questions from the audience. It's just impractical to try to do something with a large audience. But the pleasant surprise is that we have a very nice reception next door that we will adjourn to. And Professor Raff, I'm sure will be glad to answer questions. And then the final prize we have for you is a medallion from the Oppenheimer Memorial Committee. Which can be opened. If you're not wearing a bracelet on one hand. There.
I'll open it. It's heavy. Oh my gosh. Are you sure it can be open? I don't know if I can open this. Oh gosh. very heavy.
Oh, got it.
Good.
I didn't want to slap it against the lectern or something. So here it is. Before you
Oh, thank you.
show it to the audience,
gosh,
I'd like
that's
to
beautiful.
say a little about the provenance of this. So this memorial medallion was designed by the late Santa Fe artist Una Inbury. The profile of J. Robert Oppenheimer is adapted from a bust of Oppenheimer that Inbury executed in 1973. The bust was commissioned by the Los Alamos National Laboratory and was originally on, and is on display at the Laboratory Oppenheimer Study Center. The second cast is in the collection of the Smithsonian Institution's National Portrait Gallery in Washington, D.C. The third and final cast, considered the artist's copy, was obtained by the J. Robert Oppenheimer Memorial Committee and is on long-term loan at the Los Alamos History Museum. And many of us who have been to the museum see the bust. And so with that, with our extreme gratitude for being such a wonderful speaker and guest, the committee has enjoyed very much having you over the weekend. We hope we get to see you again soon.
Thank you so much. Oh,
So with that, we'll adjourn. So close it carefully because it may not never open again. You may not see it again. So please join us for the reception. Thank you again for being a wonderful audience.
Thank you so much. I love this.